You really need input from this broader, what I'd call a scientific and mentoring village, as it were, to help individuals to really navigate what's not necessarily completely straightforward. This isn't just simply doing an experiment and writing a paper. My name is Dale Abel. I'm currently at the Carver College of Medicine at the University of Iowa. I have a number of hats which I wear there. I'll tell you the different hats, but then you'll see what I'm really passionate about. I'm originally from Jamaica. The year I was born was the year when Jamaica became an independent country and ceased being a colony of the United Kingdom. I kind of always knew I wanted to be a doctor. And why do I say that? Because back in those days, you kind of only became three things in Jamaica, right? You were either a doctor or a lawyer or an engineer. So that was my ambition, I suppose. And ultimately, I went into medical school there in Jamaica at the University of the West Indies. And having learned medicine and having learned about disease and having learned about pathophysiology, I wasn't completely satisfied. There are all these books in the library on biochemistry and physiology, and there were the professors who seemed very knowledgeable. But I just didn't fully understand how did this knowledge get into these books. So I go off to Oxford, and I had not only to cope with an average temperature that was probably 30 or 40 degrees cooler than what it was in Jamaica, but I had to really come up to speed with science. And I had a wonderful mentor, Professor John Ledingham, who said, okay, welcome to Oxford. And I studied hypertension, and I noticed that many of my patients with hypertension have diabetes, and we wanted to figure it out. And so that's your project. And essentially what we did was a study in humans, where we looked at individuals who had high blood pressure as a primary diagnosis, previously untreated, and then we did certain metabolic tests, specifically glucose clamps, frequently sampled glucose tolerance tests. We took blood from them and looked at sodium hydrogen exchange expression levels in their lymphocytes and correlated that with their insulin levels. And really, we learned that individuals with hypertension certainly had metabolic abnormalities that increased their risk ultimately to developing type 2 diabetes. Because of my interest in sort of physiology of metabolic disease, and at the time, so this is now, the early 90s, when people like Jeff Flyer and Brad Lowell were starting to knock out metabolic genes in mice and get these amazing phenotypes. Mice got fat when you knocked out, or disturbed brown fat, and they were knocking out insulin receptors here and there. I said, this is what I want to learn. I want to learn how to actually take molecular insights and then model them in an organism, and of course in a mammalian organism, which was the mouse. So I wanted to learn to do that. And so my postdoc endocrine fellow project was, you're going to knock out GLUT4. And at the time, the whole notion of sort of conditional gene targeting, which we kind of take for granted now, that was brand new, very risky, but we were going to essentially make a conditional GLUT4 allele and knock GLUT4 out. We ultimately were able to delete GLUT4 from adipose tissue, and really had a very unexpected result, which has really opened up a significant understanding of the role of the adipose tissue in the generation of adipokines, or molecules that they secrete to affect metabolism. It was clear that there was GLUT4 in the heart, there was GLUT4 in the muscle, and so I said, well, let me make a cream mouse that will allow me to knock it out of the heart. And it wasn't just a whim, because another lab at the time, Maureen Sharon, had done a germline knockout of GLUT4, and besides the metabolic phenotypes, these mice had massive cardiac hypertrophy. So we did a conditional experiment, and we knocked out GLUT4 in the heart. And it turns out that paper actually came out before the fat paper came out. But they also got cardiac hypertrophy. It's like, oh my gosh, this is kind of interesting. And because of a very talented postdoc, one of my early postdocs who had trained in mitochondrial biology, and when I described to her the initial paper on the insulin receptor, she says, oh, this is all mitochondria. And I said, prove it. And she said, fine. And she did. And it turned out that then led us into a whole other area of the interaction between diabetes, insulin signaling, and mitochondria within the heart. I then became a part of a community of individuals who were very aspirational, but who also felt quite open. I have found incredibly valuable, and certainly has informed the way that I mentor my trainees now, which is to ensure that not only do they get rigorous scientific mentoring, but that they become a part of a network, both of peers and individuals who are a bit more experienced than they are, because you really need input from this broader, what I'd call a scientific and mentoring village, as it were, to help individuals to really navigate what's not necessarily completely straightforward. This isn't just simply doing an experiment and writing a paper, right? It's learning how to present yourself, how to tell a good story, how to convince somebody that they should hire you, for example. And I think those are some of the experiences that really shaped for me the way in which I've continued to approach science, but importantly, how I approach mentoring as well. My first Keystone meeting was a diabetes meeting, and I think it was in Santa Fe, and just like probably 15, 16 years ago now. And so I put an abstract, and it was probably on the knockout of the insulin receptor in the heart or something like that. And a couple of things stood out. Number one, I went into this conference. Nobody knew who I was, really. They knew who my mentor was. She was obviously very famous, but people came to my poster, and I had, hours of really engaging conversations with people who were truly interested in the work that I had to present. So that is a Keystone moment. There are, some other interesting Keystone moments which are perhaps a little bit more comical. Like, for example, this is a Keystone in Snowbird, Utah, and so the meeting ended, but the snow came the night before, and an avalanche happened, and they closed the road that went up to Snowbird. So we were all trapped in the resort, and what happened was that they had to actually put beds in the hallway and everything, because other people already come and checked in for the next meeting, right? And the people who had checked out for the meeting that was stuff ended, and so we're like sleeping in cots in the hallway of up in Snowbird, but it was okay. And in fact, there were, the meeting went an extra day, sort of, but in a much more informal way. So I think that's one of the interesting things about Keystones. You really can't quite predict what might happen, but nevertheless, people go there because they really want to immerse yourself in science for 4 days, and it kind of almost doesn't matter what happens in the end, because I think good science and good fellowship sometimes often trumps that.